It's an honor and a pleasure. And I'm looking for the remote. You're submitting me to live streaming, and I submit you to this little tune that I hope you all know, because I suppose it's an English traditional. And I've liked this a lot since I was very small. And I don't know exactly how I came to learn the words, but it reminded me of a man living in kind of harmony with nature in a garden. And what would this garden be, and the flowers in the garden, without the insects visiting these flowers? So that was my favorite hobby as a child, watching insects in the garden, and before I knew it, it turned into a profession. And during most of my professional life, I was feeling privileged, but also kind of inadequate, asking, what am I returning to society? And it wasn't until maybe some 5 years ago that I happened to stumble across the car I acquired when I was a student. And this car looks very primitive indeed, 0 levels of security, and nonetheless we took it to India and all the way back. And I realized that if I would have become a politician, I couldn't have stopped the war, and if I would have become an engineer, I couldn't have kind of I'm getting nervous because I realize this has not to do with entomology, but I couldn't have stopped the deterioration of the environment from Alpine Bavaria into Anatolia and India. And it's intriguing that we kind of get used to that, but let alone that, it puts me kind of at ease, and I realize that what we're doing, the work, is really, it is important for future generations. So that puts me kind of ease with sitting in a garden and observing insects at my age. I came to think of this when I saw my picture in the brochure. And now I'm going to submit you to some chemical ecology. I think it's not working from this distance. Or maybe. Aha. Okay. Yes. And somebody who did a lot of insect observations in the and where's the point? Is there? There must be a point. Yes. Somebody who did a lot of observations of insects in a garden in southern France is Jean-Marie Fabre, who wrote a book, Souvenir Entomologique. It's a huge series of books, and his observations are extremely precise and only rivaled by the logic he uses to interpret them. He was among the first to observe male moths flying to a calling female, pheromone communication, and he concluded that it couldn't be a chemical signal, because if it was equal to an attempt of trying to stain a lake with a couple of grains of carmine. And so he concluded it wouldn't be possible they communicate with such small quantities, and it would have to be a radiation instead. And almost precisely 100 years later, this guy, Butenandt, identified the first sex pheromone in a moth, and that was pre-GC gas chromatography times, and it took him 300,000 gallons. And he once said the invention of gas chromatography took the fun out of identifying chemical compounds, but it certainly sort of accelerated and facilitated. And later on, or simultaneously, came Schneider, who was the first to apply electrophysiology, measuring the response of insect antennae to odors. So classical chemical ecology boils down very often to extracting compounds from pheromone glands or flowers, running them through a gas chromatograph, and then asking the antenna, screening this with the antenna, and the antenna tells us that this would have to be an important compound, and chemists, the most famous and most prestigious chemist in chemical ecology is in the audience, Professor John Pickett, and then this male perfectly sees that this is not a female, but he still continues to court the glass tube because it's impregnated with this synthetic chemical. 40 years past Butenandt, Exley and Bach were the first to identify the genetic code of olfactory receptors. They started out working on mice, but the Nobel Prize lecture from 2005 was on Drosophila. That's the olfactory center in the Drosophila brain. And Drosophila has since then become a model in olfactory research. And of course, we have to submit ourselves to these new techniques. And I'm trying to take you through some of them. So stay with me. This is the head, many of you may be familiar with that already. So I'm just trying to provide an overview. This is a fly head, and this is the antenna. I didn't want to do that. Damn it. Sorry. Yes. Okay, I'll be more careful. This is the antenna, and on the antenna there are sensory hairs or sensilla. That's the olfactory center, the antennal lobe, with kind of dumpling-shaped glomeruli. That's the lateral horn that encodes learned behaviors, the mushroom bodies, and the lateral horn that encodes innate behavior. One such sensillum usually houses, again, two olfactory sensory neurons. And those neurons express in their membrane the olfactory receptors, proteins. And these proteins capture the molecules and produce make the neurons send a signal to the glomerulus. And the logic of the olfactory system has it that every such neuron expressing the same olfactory receptor sends this information branches onto one glomerulus. So those glomeruli, they bundle the information from the olfactory receptors. And an average insect like Drosophila says, again, let's count how many times I can do that. If there's 50 olfactory receptors, there are accordingly 50 glomeruli. The same thing one more time. You have the olfactory neuron in two cross sections, the sensillum in two cross sections with the two neurons. The Drosophila antenna, showing a part of it, it's like with the craters on the moon. Every such sensillum has a name. There's a map to it, and we are now looking at the AB3 sensillum. And Drosophila comes with many mutants, and one such mutant fly does not express an olfactory receptor in one of the AB3 neurons. It's perfectly identically the same as any other fly, except for one little mutation, is that this is a so-called empty neuron. And what we can do with this, using a USA driver, we can express other olfactory receptors in this neuron. And this has become a very useful technique to deorphanize or functionally characterize the binding affinity of olfactory receptors. And just very shortly, this is a regular wild-type fly. We sort of home into the antenna with the sensilla, the two neurons, the olfactory receptors, and this fly does not smell a pear ester. And then we take an olfactory receptor from any other insect. There could be a fly as well, or in this case, it could be codling moth. it's not a brilliant image, but whatever receptor it is, put this into a plasmid and then micro-inject the plasmid into the Drosophila embryo. And the wonder basically is development. Late in life, I realized one of the real wonders, of course, is development, because you can drive expression of these olfactory receptors in this specific AB3 syncyllum, in one of the neurons in the AB3 syncyllum, and voila, and now Drosophila will smell Paris. And we can use this combining the old electrophysiology technique by Schneider et al. From 57. We can go there and measure from this AB3 syncyllum. There's two neurons responding to two compounds, but they're not responding, let's say, to indolone. And then we express the indolone receptor. This one is still responding. It's just at a smaller scale, but it's still responding. There was one in the count as of now. The most important part is that we have here the reference stimulus. The other neuron is still functional, is still working. So we know, A, am I connecting to the AB3 syncyllum, yes or no? And second, does the fly work? And when this is the case, I can start to record from my test neuron, see bottom trace. So this is the molecular part, but let's not forget about the basics of chemical ecology, and this says it all. If ever you wonder about odors, it says it all, because just like vision and hearing, olfaction works at a distance. So if you're leaving the cinema at night, fish and chips, you don't have to go there and put your fingers on the grill in order to realize it's grilled fish. You smell it from a distance. And more important, so I mean, you could be wrong about the color and confound coffee and jet fuel, but you would never ever confound the odor. So these signals, they work at a distance, and they're very precise. And they are, and your system is under modulation, because you may be attracted to fish and chips once you're very hungry, but there may be times when you probably rather be repelled by it. So olfaction is long range, it's extremely reliable, and the system is under modulation. And that we need to keep in mind also with the insects. Since it's a long range signal, and we are mainly interested in working on long range signals, we use flight tunnels to bioassay the insects. And the flight tunnel assay is the ultimate proof for that it works. And part of a very important part of the flight tunnel assay is the odor sprayer. That is a machine, just like the GCAD, designed by Heinrich Arn. It lets you allow compounds in dilution. Wait a minute. It lets. What did I say? You can spray compounds in dilution at known quantities and known chemical purity. And the beauty of it is because if you would sort of apply things to filter papers and rubber, you need copious amounts of compound to make them work. But with the sprayer, it's enough. You collect headspace for a couple of minutes from vinegar, and then you can take this headspace collection into a solution, look at it on the GC, check out what's there, quantify the amount of, say, acetic acid in this headspace collection, and then spray it. And then, of course, sort of compare live vinegar as such, spray the vinegar headspace collection, and find out, well, it's equivalent, and then we can start deboning and dissecting the chemical components of vinegar. So we've been doing that for a while, and we're studying the components of fruit attraction. And yes. And the other component in the olfactory, two important components, of course, in the olfactory world are food and sex. Basically, sexual communication makes sense only in the light of reproduction. So obviously, subserves reproduction, so food and sex are tightly interconnected. And this image tells us that fruit flies, like a banana, that's a citation, unfortunately I didn't make this up myself, but it also reminds me of how rewarding and instructive home cooking can be, especially when you manage to maintain sizable fly populations in your home in spite of your spouse. And then when you eventually sort of get down to producing yeast dough, you realize that the flies start homing in on the dough that doesn't contain any fruit. So that tells us that the fly attraction to the fruit must contain a microbial component, and that's exactly the case. A sterilized grape attracts fewer flies than if you inoculate this grape with yeast. But most interestingly, a petri dish with minimal medium inoculated with the same yeast produces the same amount of attraction and the same amount of oviposition. And the fact that the flies do survive, they don't survive on minimal medium, but they do survive on sterilized grapes, is just because you can never ever, for a 2-week rearing, produce a grape that is absolutely sterile. Even if you use sterile flies, there will always be some bacterial infestation of some sort. So now we know the flies, yeast is an important component of their life and their long-range attraction, but we're missing the sex component. And most precisely 10 years ago, we were given a certain amount of money and we could do whatever we wanted to, and we said, okay, let's check out whether they're using a long-range pheromone. And we started doing that, and we're asking, well, is there more precisely a female-produced pheromone that brings the flies to the source from a distance? There is a compound that has been identified in '85 by Bartelt, CBA. Most of the high-class papers today don't cite that work any longer because it isn't good enough. So they refer instead to the reviews in Nature Neuroscience and other journals. But in '85, Bartelt identified a male-produced pheromone, but this pheromone doesn't attract the flies over a distance, and it's active only during courtship. So and it's shared by many other Drosophilids, which means it doesn't convey species specificity. And yes, so we started out collecting fly odors, males and females, into glass vials. And at some stage, when working with my research associate Paul Becher in the lab, I realized I had confounded the male and the female vial, and as usual, not marked it. And he says, don't worry, that's the female vial. And I said, how come? How do you know? Can't you smell the difference? Well, I couldn't, but he could smell the difference between the males and the females. And that was a nice starting point. And earlier in life, we have been developing pheromone-based insect control methods in collaboration with the wine growers in southern Germany. And therefore, we maintain good connections to them and their taste panel. And that is the most prestigious wine tasting panel in Baden, which found out that, yes, even they can distinguish between the odor of a male fly and a female fly. That, of course, has nothing to do with Drosophila, but it sort of, yes. And then we have this TV show in Germany, and you can submit bets to it. And the show we were aiming to get into was where a policeman could spot the tax plate on a number plate on a car passing at a speed of 100 kilometers. He did it 2 out of 4 times, so he failed the bet. But it was anyhow, there was vision. The other was hearing. There was a plumber who, blindfolded, could discriminate between the 12 most frequently sold water closets through the sound of when flushing them. So I felt it would have been a wonderful thing to bring the flies in and smell the difference between male and female flies, but we weren't admitted. So we couldn't take the shortcut way to fame, and had to resort to empirical science instead. And since this is live streamed, and that's the only reason, that's the only excuse I have for dimming out the compound name, because it's not published, and we're undergoing the regular hate review process in the third journal now, and so it's not for my sake; it's for the sake of my coworkers. And it probably doesn't make a difference, because it's anyhow, most people have so much to do, so it's anyhow safe. But Sébastien Le Breton knelt into the chemical analysis of fly headspace, and you see the violet trace on top, that's a female, and you see a number of peaks that are not present in the male chromatogram underneath. And especially peak number 5 sticks out as something particularly interesting, and it's not there in the next closely related species, Drosophila similans. And then I skipped the biosynthesis because that would tell you what it is, and we go instead into the single cell screening on the antenna. Like I said, there is a map of the sensilla on the antenna. And we can screen, OSN class is really not precise. I just realized it should say sensillum type. And we go through all the sensilla, and we find out it's the AB9A that responds to the compound. And yes, there was a small response in the AB4, but a GC single cell run shows us that the AB4 responds to an impurity, trace amounts of a chemical impurity in the synthetic batch. So it's really the AB9A that responds. And the nice part of the really rewarding part of Drosophila research is that it's kind of, at times at least, logical. And the logic of the olfactory system has it that we now know that it's the AB9, in former times, it would have been enough to identify the compound, make a synthetic copy, and bioassay it, and that's it. And that is no longer sufficient. Nowadays, people are asking what's in sensillum, and especially what receptor is responding to it. And that, of course, makes a huge difference whether there's a receptor that is specifically tuned to a compound or not, or whether maybe a range of receptors all respond to it. So inside one sensillum, the AB9 sensillum, there's 2 neurons, and obviously they're expressing 2 olfactory receptors, so we still don't know which receptor. We just know it's in the AB9A. And the next is a technique called functional imaging. So through Drosophila, the system is under genetic control, and it can express fluorescent proteins in selected neurons. And on top, you can do this overall. So this is GFP is expressed in the entire antennal lobe, and the lower is expressed only in a group of neurons. And those are output neurons. Those are the PN neurons that connect the antennal lobe to the higher brain. And the overall result of this imaging shows us it seems to be the D glomerulus. At the antennal lobe level, there's still some cross talk between the glomeruli. There's local interneurons, so the image there is not as precise. But once you know this technique, and especially the imaging, the output neurons to the lateral horn shows us that's the D glomerulus. And the D glomerulus was earlier known to respond to linalool, but it also responds to this to the scent of the fly. And now, well, and you see this is a map of the antennal lobe with all the different glomeruli that is all already known, and the D glomerulus is there, and it's known that the OR69A is expressed in the neurons connecting to the D glomerulus. And then almost looked like that's it, except that usually there's only one protein expressed in the olfactory receptors. Here we're dealing with the spliced olfactory receptors, and that's kind of unusual. And most people said, the spliced receptors, what can they really mean? Usually it's redundant. But I mean, nonetheless, there is two areas coding, two sequences coding two different proteins. But since they're driven from the same exon, they are expressed in one neuron. So the situation is that there's a difference between the 69A and in other olfactory receptors in that these neurons express two proteins, and the glomerulus, which collects the information from the 69A expressing neurons, is sending on a mixed message to the lateral horn. Have I lost you, or are you still with me? Because I need especially the evolutionary ecologists to listen, because hopefully I sort of convince you that it's anyhow interesting. So we screen the OR69A, the AB9A scintilla in wild type flies, in Drosophila population from Africa, which is reproductively isolated from the cosmopolitan European Drosophilas, and in Drosophila simulans, which is the next closest relative. I'm kind of halfway through, and that's sort of, I'm still holding. And you see, there's kind of this two peaks of the receptor responding to two groups of compounds. That's the wild type. And now, of course, the one that happens, we express these spliced OR versions one by one and together in empty neuron flies. So we have the A and the B splice variant, and both together. So both together produce a appreciably similar image, except for this one compound. Hard to tell exactly why this is responding less, but of course it's electrophysiology, and we're counting the spikes and so on, and there's room for some variation. But in principle, it shows again this dual, these two peaks, and you realize that those two splice variants, they don't produce they don't respond to the same set of compounds. So this one is responding mainly to yeast, volatiles, and that's terpenoids. So terpenoids are not only produced by plants, but also by microorganisms, and this is rather sort of tuned into the pheromone. Yes, and then if you would know the chemical structure of the pheromone, you would say, well, what has this to do with linalool? It's completely, structurally completely apart. But conformational analysis shows that the two ligands up here for the A variant, terpineol and tell me, the linalool, of course, they overlap appreciably. That's sort of understandable, but even this compound and carbone overlap, and it's sort of it's kind of it makes sense that these produce a response from the same receptor. Since these splice variants are available in a number of related species, this is an absolutely wonderful substrate for structure-activity studies. And very close to Lund, it's the MAX 4, the largest synchrotron facility, has just opened up. And of course, the ambition would be to take this into the very high-power microscopes and study structure activity and conformational analysis even in the proteins later on in life, or for the next generation. Meanwhile, my hobby is the wind tunnel. And I did find, and every time I find a student who is willing and able to run a wind tunnel experiment, that is the major achievement, because it takes so much discipline and so much understanding for the insects to fly them in the wind tunnel. So Felipe from Corpoica in Colombia, Bogota, did wonderful wind tunnel work, and he shows that this compound attracts both males and females of the fly. So it's a female-produced compound, and it attracts both sexes. That makes sense because this AB9A circuit is outside the fruitless circuit. The fruitless circuit in Drosophila is known to produce a sexually dimorphic response. So 69A is outside the fruitless circuit, and therefore it's producing a sexually isomorphic, but it's producing the same response in both sexes. When we look at linalool, which is the ligand for the other splice variant, and linalool, that is a yeast compound encoding food. It attracts also simulans, but interestingly, when you blend the pheromone and linalool, it only attracts melanogaster, but it doesn't attract simulans anymore. So there's an antagonism which kind of reinforces the idea that food and sex collaborate. And then, of course, what I failed to say, I should have done this earlier, when showing you the picture of the two splice variants feeding into the same neuron. During phylogenetic divergence, when the splice variant appears, of course, since they're feeding into the same neuron, it isn't entirely independent. If it was a regular gene duplication, the newly formed receptor could start all the olfactory receptors are under very strong selection pressure because there's only 50, and the flies don't carry around receptors that are superfluous. So they're all under fairly strong selection. If a receptor comes into being through gene duplication, it's free to adapt to or start to sense any volatile compound. But since it's the splice form, the newly formed splice form feeds into the same neural circuit, it must follow the same thing. Not knowing what was first, pheromone or yeast compound, because we don't know phylogenetically what the original variant was responding to. We will find that out very soon through going back in phylogeny to the older species. Assuming it was the pheromone that came first, so the splice variant could choose to respond to another pheromone compound and just enforce the pheromone reaction, but the fact that it tunes instead to a yeast compound or vice versa is a wonderful demonstration of how tightly, intimately interconnected food and sex communication is. Yes, and then because it's Drosophila, that's sort of part of the exercise. You need to knock out the receptors in order to really show it's the 69A, and that's what we do here using canton flies. Those are lab rats. We use instead otherwise Dolby flies, which are collected from Sweden. And then we express tetanus toxin and to knock out specifically the OR69 receptors. And then that's when we write papers about that, when I start to wonder, is it for real? Does that really work, or is it all a hoax? Am I being submitted to some kind of experiment? Someone is watching over me, how I do when we write up this stuff. But it seems to work. And now back to the red and green, to this dual thing, sex and food. Yes, pheromones, as we saw it, they are subtle, very powerful, subtle. The green stuff is kind of more abundant and maybe less distinct, maybe, who knows. One thing is for sure, pheromone is always released into an atmosphere of plant volatiles. And when we once wrote this in a paper a couple of years ago, one of the reviewers asked us, can you soften up this argument? And this is just because olfactory physiologists have chosen to work in lab space on sex pheromones where there's no plant volatiles. So we could go on for a long time just testing pheromones, but pretending the plant volatiles aren't there. But for any insect herbivore, the male will never ever perceive pheromone as such without a background of plant volatiles. That's very important. If ever you doubt this, think of this dog, because it's not only the definition. If ever you're insecure about the definition of pheromone, it's a compound produced by an organism released to the outside, where it's perceived by another individual of the same species, where it elicits a behavioral reaction. And the behavioral reaction is sex-specific, because it's totally different whether it's a male dog or a bitch in heat that releases the signal. And it's always, most of the time, it's released into a sea of plant volatiles. And we have ourselves released pheromones into the environment for a very long time. A wonderful title, odors instead of poison. And we did even found a company and register commercial pheromone products just 1 year before they deregulated the most widely used insecticide. That was good thinking. That was exactly when we started to work on Drosophila. And we never considered that we were releasing these synthetic pheromones into an atmosphere of plant volatiles. Lucas, there is the mechanisms of mating disruption. Is there one discussing the role of plant volatiles? Now there is. But it's hard to believe that it hasn't been around for much longer. Then another important issue, and I'm almost done with, I'm touching the end of it. When I say green volatiles, make sure that you know what you think. There's different types of plant volatiles. They're not all green. And there's, in ecological literature, there's the so-called bad mother syndrome. That is, that females, at the expense of laying eggs, spend their time buzzing around the flowers and feeding themselves. And I once had a very religious student, and I wanted to make him write an introductory paper on the bad mother syndrome, and he just plain refused. But instead, he gave us a most enlightening insight. That is, unmated females respond to floral volatiles in order to feed themselves, and as soon as they're mated, they turn into green volatiles, and the entire olfactory system changes, and this helps to discriminate between food as a plant volatiles that signal adult food and larval food. That's also important to know. What's the significance of the plant compounds you're working with? Another proof for the strong interconnection between pheromones and plant volatiles comes from neurophysiology. That's again functional imaging, optical imaging in the antennal lobe. In the male antennal lobe, there is a huge thing called the cumulus. That's an enlarged glomerulus that is just treating pheromone signal, and that's the response to the sex pheromone of codling moth, pheromone. And then there's a well-known, identified in 2000, I think, or 2001, a strong plant volatile, which is the strongest known attractant for codling moth. That's pear ester. And the trouble was always with pear ester that it's produced by pear and not apple, which is inconvenient because the codling moth is mainly attracted to apple, really. And nonetheless, but it's an important, behaviorally important compound. And when you blend both of them, it sort of becomes a kind of a Fukushima meltdown, a very strong synergistic effect, showing again the intimate connection between those two types of modalities. And totally unexpected, another proof comes from an antennal transcriptome of codling moth, and I was trying to persuade the editor of a journal the other day trying to argue that we can start reading those transcriptomes, and on the basis of the information we have about olfactory receptors, we can start to reliably interpret them. And of course, there's no proof in the transcriptome by itself, but it affords reliable hypotheses. And one of them is, if you compare that's a part of the larval and the female and the male transcriptome, that's the co-receptor that's present everywhere. You realize that the larvae, that's the expression, the brightness is the expression level, and the bright spots here in the larvae show you that the larvae are kind of different from the adults. That's not surprising. Then part of the know-how for olfactory receptors is that you can reliably distinguish between pheromone receptors and the so-called regular olfactory receptors. So, and among the clade of pheromone receptors, you will immediately, what pops out is, well, first of all, it's the receptor for, it hasn't been deorphanized yet, but it's most likely, very, very likely, that this is the pheromone receptor for the main pheromone compounds. It's very likely that this is the pheromone receptor for a pheromone antagonist, because this is the OR which is most highly expressed in the sibling species, which uses codamone acetate as a main pheromone compound. And most intriguingly, of course, there is a pheromone receptor expressed in the females, and that is, of course, a prime source of investigation. And I'm not making it up, but well, this is the regular interneuron thing, and you see that OR3, CPOM OR3, is responding, first of all, to pair ester. And that sort of explains also the antennal lobe synergism, because we know that pheromone receptors, they kind of branch to the same part of the brain, so it's adjacent glomeruli that produce this synergism. So on a physiological and genetic basis, we confirm the behavioral experiment, which is kind of our mission. That's really the very core of what we would want to do. And then this all, I think, doesn't make sense. Well, it makes sense to us like it is. It's good enough, but I mean, of course, the next step is to see this in a slightly broader context, and that is evolution, obviously. I would welcome any comments from the audience on that. I stumbled across 3 papers a very long time ago. The quite famous Patterson, which has been kind of ignored by some, because he contradicts the isolation concept. And I remember first reading him, I was not entirely in favor of his ideas, because he basically says, during the evolution of mate communication, species, the only thing they want to do is optimize interspecies sexual communication, but they really don't care about other species around. And any reproductive isolation effect is just a matter of pure coincidence. And I, at that time, felt that those pheromone antagonists, like we saw it in codling moth is expressing a receptor for a pheromone antagonist. That is, when you have both compounds in a lure, you don't catch any codling moth anymore, because it's the pheromone of another species. So I took the presence of pheromone antagonists, so the fact that they respond even to pheromone antagonists, I took that as a proof for the isolation concept. But I'm not sure any longer. I think it may be an artifact due to the architecture of the olfactory system of some sort. It's not necessarily, I'm not quite sure that there is selection, that they would spend time and energy to really produce, put much effort and attention to pheromone antagonists. I'm not quite sure about that. But definitely it's important, as they try to achieve effective mate communication, and importantly, this mate recognition refinement takes place in the natural habitat. And Patterson, I think, was among the first to realize that, and accentuate that. And another very famous, very frequently cited paper, which I found like 3 weeks ago, is first time ever. It's Endler, it's the sensory drive hypothesis. And I think I ignored it earlier mainly because it's used to explain the color patterns in cyclid fish in the tropics. The males have sort of bright orange chins that remind the females of citrus fruit that they're hunting for that falls into the rivers. And that's but otherwise, the sensory drive hypothesis it refers to any bias in the direction of evolution of communication. And one important bias is the local environment, partly because it may affect signal transmission. So you need to have signals that sort of, for example, olfactorily speaking, point out of the sea of other compounds. Adaptation to local environments also with respect to, yes, food sources for the larvae, and also, of course, how well the signals match the existing receptors, because when the splice form or any duplicated olfactory receptor comes into being, it's already pre-shaped. It doesn't sort of, it's not completely open to everything, but there's already a substrate. And evolution doesn't create it de novo, but it just works on whatever is possible within the substrate. So that would be another sensory bias. And last but not least, a more recent paper coining a catchier and easier to remember term, the so-called magic traits. And the magic traits basically is reshuffle, according to what I read, it's basically a reshuffle of the previous two papers, but notably you find the term the collaboration between natural and sexual selection, and that's sort of easy enough for me to remember. And so this, in principle, we aren't there yet, but this olfactory receptor has the potential to be a speciation gene, because it conveys species specificity, species-specific sexual communication in the fly, and it's combined with larval food resources. So, and yes, it's unpublished, but I mean, since it's a new story and we are all data addicts, maybe it wouldn't make a big difference to you whether I talk about old or new experiments, but it does make a difference to me. And I hope, was that kind of interesting, or are you flat on your stomachs? And this is the result of the gardening. It took me 2 weeks. Thank you. I think we can probably.
We'll have to do it otherwise. James and I practiced with this yesterday. Okay, are there any questions? Right, John?
Thank you, Victor. The issue of pheromone divergence is really crucial, I think, because obviously if we do, as Mary Hamilton said yesterday, manage to use chemicals and pheromones, particularly in crop protection, more than we are at the moment, perhaps by delivery from the plant itself, which is obviously what I'm interested in, we will need to contend with that. I mean, my view is that it's not a problem as it is with insecticides, because you can stay up with what the natural system is using. You've got an example there of looking at the actual mechanism. But the mechanisms that are in the literature at the moment look like non-synonymous SNPs that arrive at somewhere around the active part of the final synthesis enzyme and cause a change in the compound. splicing mechanism that you suggested?
I'm not 100% sure whether I follow entirely, but I mean, what I'm trying to say is that if you have a regular duplication event, so sort of a new receptor comes into being and is possibly, say, expressed in another sensory neuron, then this new receptor is free to evolve in whichever direction. So the intriguing thing here is that the evolution of a spliced receptor is restricted to a behavioral theme in the first place, and secondly, it's kind of it's kept together during phylogenetic diversification. The Drosophila system, there's a whole species complex that is with known genomes, and there we can sort of there's a timeline through this due to the fact that they stick together. In the other receptors, you don't know for sure when they came into being and for how long they have diverged, and they are submitted to different selection pressures, and it's interesting as well. But I mean, this is a particularly interesting case because it's behaviorally very important, and we sort of can trace the changes in these two splice variants through diversification of the Drosophila melanogaster subgroup. But I mean, that's maybe not entirely what you were asking. I'm not quite sure.
Well, we don't need to digress at the moment, but anyway, you're going to investigate that, as I understand it.
We have the empty neuron flies arrived as of yesterday, well, a week ago, and now, as you know, we ran out of funding. And now we are there we have everything except manpower. So but that's a different story.
I wonder if the receptor genes are clustered in the genome of the Drosophila, and whether you see maybe positive selection.
The clustering, that is another sort of intriguing part of this spliced thing, is that, of course, before people with the magic traits, I don't know whether you're referring to kind of pleiotropic effects of genes belonging together somehow, because when talking about this collaboration between sexual and natural selection, if it's two different ORs, the first thing you're asking, are they somehow interconnected in the genome? Is that what you're asking? Yes.
So I wonder if many of these two introns are together in the genome, like cluster of families.
So if it's independent olfactory receptors, they might be interconnected through some mechanism, which in most of the cases we don't know today. But there are such mechanisms. But that's sort of for at our level, it's kind of elusive. We are not at this level that we could sort of work on such effects, such phenomena. But I mean, we know with the spliced receptors that they belong together. And that's sort of another, that facilitates the situation. Because before, between pair ester and collimon, the calling moth sex pheromone and pair ester, that is also something that is kind of belongs together. And we found now the pair ester and collimon olfactory genes in other species. And you're of course asking, is that somehow interconnected? But that remains elusive. And that's another reason for, I'll spend the rest of my life investigating this spliced olfactory receptor. It's not an entirely positive aspect. I feel it's slightly frightening, but I think I would reduce it to 69A and B.
When I took insect physiology, rather simplistically, I was taught with pheromone coding, going from the receptors to the sort of taught that it was lateral lines, information transfer from the receptors to the macroglomerulus.
That's the dumpling.
Cerebrum and then to the motor neurons to cause behavior.
And unfortunately, I'm not in the position to sort of give you a general answer because I'm not a neurophysiologist either. So unfortunately, I can't really tell. But sort of, if I was, I sense that. Well, you have these labeled line things, and a labeled line is like one receptor, and it goes right through to the lateral horn, and they start flapping their wings. And the concept with the plant volatiles for a long time it has been, and I didn't want to sort of bring this up now at this point in time, but I mean it will have to be said at some stage that we earlier submitted our poor insects to hundreds of GCAD runs, and I spent many years in its complete crap. And why? Because you're screening the differences. Heinrich Arndt designed the GCAD to be used with heterospecific male antennae detecting pheromone compounds that are produced at minor pheromone compounds that are produced at very small amounts. So the basic thing was you have a pheromone extract with a couple of pheromone peaks, and you're insecure about a tiny little peak in the trace, and then you take in the antenna of another species for which this compound, this preceding compound, is the main compound, and then you sort of identify it. Or at least with pheromone, because the female gland contains few compounds. It's a long answer now, sorry. But I mean, that works. The GCAD works well with pheromones. But it doesn't work with plant volatiles. Why? Because, as we all know, the plants don't care about the insect response to the plant volatiles. The plant volatiles are produced at copious amounts, at very large amounts, and the receptors, all of them, somehow, if you sort of load the antenna with alpha-farnesene, there's a couple of receptors that will eventually sort of respond.
Yeah, that's the wrong answer, isn't it? Because you're then looking at what you can do.
Yeah. But that's the GC. But that's what the GCAD gives you.
You've obviously got to put the sample on at a relevant physiological level. But in answer to that point, I mean, it depends which the volatiles are, because although some appear to hit at a fairly high level this kind of general response, some are extremely sensitive. And from your own group or colleagues in Sweden, you have the wonderful Geosmin paper in Sept, where there's a very specific recognition of an antagonistic compound that relates to whereas you can get very selective and not very selective. So I don't think you can say that the tools are not suitable. They're misused, certainly.
I'm pretending. I'm using words which don't convey really my feelings. The way I see it is entirely different. It's amazing that chemical ecology has dealt with a male response to the female pheromone forever. And interestingly, many of those olfactory physiologists are colorblind in addition. And I find it interesting that out of a sudden the female response would be entirely different. The males have a highly sophisticated and very sensitive antenna that is tuned to the pheromones, whereas the females respond. I'm sorry, I'm not contradicting. I'm just saying the females are thought to respond to a blend, a species-specific, plant species-specific blend of huge amounts of compounds that occur in every plant. Why would that be so different? I think that the Perester example shows that it's exactly the same in the females. There's tiny, tiny little peaks of compounds that are rather species specific, and the female antenna is almost or just as sensitive as the male antenna, and they don't care about alpha-farnesene and beta-caryophyllene, but they tune into tiny little signals that convey specificity, and that's Perester, for example, in calling moth. And it's maybe not complete, but it's specific. I'm sorry, it was too long an answer, but I don't see a principal difference between there's label lines when it comes to compound. And I didn't want to contradict you either. I'm sorry.
I think we'll call that.
You realize this now you have to stop me somehow.
Discussion over coffee. Thanks very much.